we are going to do things a little bit differently. We're going to start here in chapter 14. I'm going to post a YouTube lecture for the end of chapter 13, but I think it will make more sense to try to get through chapter 14 as a unit instead of breaking it up into two pieces. The first slide, and we briefly review cloning at this point. And so cloning is something we've talked about before, and we talked about both reproductive, Sam, and therapeutic cloning. And, so we won't go back over that particular topic, but we do want to talk about gene cloning today. And so gene cloning is a term that we use, Sam, to describe the process by which we make many copies of a particular gene that is of interest to us. And we do it in the following manner. If I can advance the slide, then we will see. Here we are. So we do it by making essentially what's referred to as recombinant DNA. Kyle, recombinant DNA is DNA that comes from two different sources. And because DNA is the same in all living organisms, Matthew, this is possible. So what we're doing on this slide is the following. We have a human cell, and we pull out a chromosome from the cell. So I'm pointing, as I think all of us can see with the cursor there. If I go over to the right-hand side of the screen, I see a bacterium. In the bacterium, I have just the chromosome, which is a circular loop all wadded up there, and I have a small plasmid. The plasmid is a small loop of DNA that contains 10 to 100 genes, Ethan, that are usually for extra things. And so what I'm doing is pulling the plasmid out, and I'm cutting both the plasmid and the human chromosome with something referred to as a restriction enzyme. And we'll talk about restriction enzymes on the next slide. But essentially, Matthew.
This is 14?
It is, isn't it? Yeah. Yeah, it is. Yeah, it's similar to what we talked about previously. But we didn't talk about this process, did we, Matthew?
No.
Okay. Yeah, it's a good question. So what we do is take the gene for human insulin and we place it into the plasmid. And so here we have some recombinant DNA. So the light pink is the bacterial DNA, and the red here is the human DNA. And so we then, interestingly, Sam, take this plasmid, place it back into the bacterium, and the bacteria, every time it reproduces, then it copies its own DNA, but it also copies the human DNA that's in that plasmid. And so, Kyle, a bacterium can reproduce every 15 minutes, E. coli that is, if you give it optimal temperature and all the food that it needs. So you can leave at 5 o'clock one afternoon, Ethan, and come back, Brooklyn, at 8 o'clock in the morning, and you have millions of copies of this gene. So it's a good way to clone a gene, to make multiple copies of a gene, Sydney. And to do that, we need something referred to as a restriction enzyme. So a restriction enzyme is an enzyme that cuts DNA at a particular point, Spencer. So if we follow the diagram here and follow the cursor on the diagram, what we see is that this particular restriction enzyme recognizes an AATT sequence, but it cuts the DNA not in like you would cut it with a knife, like right here, all the way across, but rather it cuts the sugar phosphate backbone here, and then it cuts it over here. And so it leaves these overhangs that are referred to as sticky ends, sticky ends. They're called sticky ends because they will form hydrogen bonds with any complementary base. This A will bind with this T, this A will bind with this T, and so forth. And so you can mix and match DNA in using this manner, Sam, using this technique. You can have human DNA on one side and you can have cow DNA on the other side, and they'll glue back together, Matthew, essentially because there's no difference in cow DNA and human DNA aside from that difference in order of bases. So you do need, of course, a DNA ligase enzyme, Jadlin, to seal that sugar phosphate backbone like we've talked about previously. And then you've got to get we skipped over this part earlier, but then you've got to get the bacterium to take up the plasmid, but that's easily accomplished. And this is not really high-tech science. I mean, maybe it's high-tech science, Sam, but it's something we can accomplish in introductory biology labs. So it's not something that's very difficult to accomplish. You can ask the question, why do you want to do that? Well, as we'll see later in our lecture, oftentimes you want to copy genes to make a product. And so if you know someone that takes insulin or know someone that has to take human growth hormone for some medical issue, then the product they're getting comes from bacteria. It's by far the easiest way to do it. All of the insulin that's taken by diabetics is made via this process. Thoughts or comments there? Either from our in-person or Zoom participants? All right, so we now want to talk briefly about complementary DNA. Complementary DNA rarely actually exists in the natural world, but it's mostly a laboratory procedure. And so what is done is to make complementary DNA is the following. Here we have messenger RNA, and we take an enzyme that's referred to as reverse transcriptase, and we use it to copy the messenger RNA into DNA. The DNA that's copied is referred to as complementary DNA because, Kyle, it's complementary to the messenger RNA. Now, this enzyme, reverse transcriptase, comes to us from a very small number of viruses, such as the HIV virus that causes AIDS in human beings. It's essentially taking the central dogma backwards. We always go from DNA to RNA, but this is going from RNA to protein. The HIV virus, Ethan, uses it when it enters an organism, when it infects a human being. The virus is RNA, but then it copies itself to DNA and then inserts into the human cells and remains there. And similar to herpes, there's no way to cut that back out. COVID is also an RNA virus, but it doesn't become part of your DNA. It just infects you for a while, and then if you're successful in overcoming it, it goes away. But complementary DNA is a useful technique for this particular reason, because we cannot copy RNA, Sam. We can't copy RNA. We can copy DNA. It's very common to want to measure the RNA that's found in a cell. What does looking at the RNA in a cell tell us that looking at the DNA does not? Well, what's the difference, Kyle, in the DNA found in a liver cell and a skin cell? It's exactly the same, isn't it? So the DNA in every single cell in the body is exactly the same. But would the RNA be different in those cells, Matthew? It wouldn't. So different parts of the DNA are red in each cell. That's why different cells are different. So it's very common to want to look at the RNA that's found in a cell, and this lets us do that, making complementary DNA and then copying it so we can see it. Now the next slide is the topic of our lab this particular week. And so there's a pre-lab lecture that's posted for that. However, I do mention in that pre-lab lecture, Sam, that I don't really talk about the science of what we're doing. It's more just, you point to this, click on this, here's how you do the lab. And so I do strongly encourage you, as I said in that pre-lab video, Jalen, I encourage you to look at this video and make sure you understand PCR before you can attempt that lab. So PCR, which stands for the polymerase chain reaction, was invented by Kary Mullis in the late 1980s. Kary Mullis is an interesting guy. He's a Nobel Prize winning scientist, and I've wanted to read his book for a long time. His autobiography is called Dancing Naked in the Minefield. It's pictured up here on this slide. Kary Mullis is something a bit unique in terms of Nobel Prize winning scientist, because he's the only Nobel Prize winning scientist, to the best of my knowledge, that has ever seen a glowing alien raccoon. And so did he really see that? Well, I don't know. It doesn't seem super likely, but it's hard to rule everything out, Kyle. Now, he did struggle, he has had some issues with drug use at times, which is not super common among Nobel Prize winning scientists, Ethan. So it's quite possible that it's tied to that. But he really is. I don't in any way mean disrespect towards him. And he did pass away, believe it or not, last August, which disappointed me greatly. Kyle, I always wanted, he's not a very popular Nobel Prize winning scientist. So I always thought, well, maybe if I send him an email, then I could say, hey, if I fly out to California, could I buy you lunch and just ask you some questions? But it's not going to happen. So I didn't even know he was in poor health. So I really want to read that book, but he was called as a witness in the first O.J. Simpson trial because DNA evidence was so new, Kyle, at the time. That would never happen today. And Simpson could afford to hold a Nobel Prize winning scientist as a potential witness, but he never took the stand. And he got a little bit uneasy or a little bit sensitive about what people were saying about him, as perhaps any of us would. And I found this quote regarding that issue at the time. And so I thought it was a good quote. He says that it wouldn't matter if I go completely nuts or I was nuts when I invented PCR, Jadelyn. It still works, and it's sound science. And so it is. And he is I think I encourage you to Google his Nobel Prize acceptance speech and read it. It's really interesting. I'm not sure about the book. I haven't read it yet. I do want to. I'll let you know after I read it what I think about it. So essentially, PCR is this. If you remember nothing else, and you'll want to remember a lot, PCR allows us to copy DNA. And it really revolutionized biology because up to this point in time, Sam, we just didn't have any way to copy DNA. And how big is DNA, Matthew? Pretty small, right? So you can't just squint your eyes and see it, right? You can't take a magnifying glass or, for that matter, a microscope in the lab and look at it. It's just too small. And so what this PCR allows you to do is copy particular pieces of DNA and visualize those. The PCR uses the following method to copy DNA. Now, this is very important for your lab. It's also outlined in your lab, and I think I briefly mentioned it in the pre-lab video. So we go through these cycles. I'm pointing now with the cursor up on the screen. So here's the DNA we want to copy. The first thing we do is heat up the DNA to denature it. By denature, we mean split it into two different strands. And so we've broken what type of bonds, Emma, that hold the two strands of DNA together?
We've broken what type of bonds?
What type of bonds hold the two bases together? A type of, it's a weak bond, the weakest of the three bonds that we talked about. It is hydrogen, but good job, Emma. Sorry, I was afraid for a minute we weren't going to get you there. So hydrogen is perfect. So we break the hydrogen bonds, and the temperature that we heat it to is most likely, Ashley, 95 degrees Celsius, maybe 94. Now we have the two separate strands of DNA. So now, Ashley, we cool it down, and this is called our annealing temperature. It allows the primers, shown in green here, to bind. A primer is something that tells the DNA polymerase where to start copying, Jadelyn, where to start copying. The primer tells the DNA polymerase where to start copying. Now, I think your lab uses an annealing temperature, Ethan, of 50 degrees, which is kind of low. But it's an annealing temperature of 60 or 58 would be much more common. Then the last part of the cycle, and all these just take a few seconds, Sam, is we heat the DNA up to 72 degrees. And we heat it up to 72 degrees. That temperature is ideal for the DNA polymerase to copy the DNA. And there it goes, adding bases, as we have talked about in the DNA replication chapter. So remember, this is all in a test tube. You understand that, right? So none of this is natural. This is all in a test tube. So Ashley, after we complete one cycle, how much more DNA do we have than we started with?
Twice as much.
Perfect, Ashley. That's excellent. So we double the amount of DNA we have every time. Very good. And this means in 2 hours, we can have 1 million copies, and we can actually see 1 million copies of DNA. And so remember, your lab only runs it for 1 minute and 45 seconds. They didn't want to make you sit there for 2 hours or come back later. But that's not realistic. They tell you that because you have to run through many of these cycles. Now, one last thing to add here, this DNA polymerase, interestingly, it's a special polymerase. You cannot use a human DNA polymerase because this is all in a single test tube. And when you heat it up to 95 degrees, you would cause denaturation, Ethan, of the human DNA polymerase. And so a DNA polymerase from a bacterium called Thermus aquaticus found in the hot springs in Yellowstone National Park is used. So this is an organism that's well adapted to these very high temperatures, almost boiling. And so its DNA polymerase does not fall apart when you heat it up. And so it's referred to as TAC, T-A-Q polymerase, an abbreviation for Thermus aquaticus that has become popular. So TAC polymerase is a key part of this overall process. There's Mullis, and here's the role of primers. Now, I just want to emphasize to you before we leave this, primers allow us to, they tell the DNA polymerase where to start. So on the screen, we have a cytochrome B gene from a salamander, and we wanted to copy everything from where I'm pointing here, this cytosine, all the way down to here, this cytosine. And so between those two cytosines, there are 958 bases. And so to copy that, we had to make a primer that bound to the green area and a primer that bound to the red area. And remember, the joy of making primers is this, that each DNA sequence is different. Mine differs from yours a little bit, but especially as you go from one species to another. Like, for instance, a primer would bind to a human gene, but not to a gorilla gene, but potentially, although we're pretty tight there, pretty closely related. So whatever the case is, you need to figure out what gene you want to amplify and make primers. Primers cost about 35 cents a base pair, Sam, very, very little. And the primers are 19 to 24 bases in length, and so you come up with a primer that will bind to your target DNA and make copies of your target DNA. Because PCR, I do want to be clear, you're not copying everything. There's no question about that, right? You're copying at the most 2,000 bases. How many bases are in your genome? Well, 3.2 billion. So you're not copying anywhere close to your overall genome. And remember, this process is used just everywhere in the world. I was trying to think of some examples as I was talking here. So one interesting one is this. So now when there's a shark attack, how do they tell what type of shark it was? They take DNA from the wound and amplify that, and they can tell the species of shark based on the DNA that the shark left behind. Ethan, in your potential career field, trying to apply it there. But if you want to identify any animal, you can identify that animal based on the DNA. So here's one example. Did I tell you this recently? So I can't remember if it was you or another class, Kyle. There was a hunter that said they shot a cougar with an arrow. And there are not any cougars in Kentucky, unless someone lets one go. But the arrow went all the way through it and stuck in a tree. And so Fish and Wildlife was interested in that. They took the arrow, removed blood, extracted DNA, amplified it using PCR. And what did the hunter actually shoot? What did he shoot, Ethan? Bobcat, right? It was definitely a bobcat. So you can use this for all sorts of applications. The next step of PCR is this. You want to analyze the segments that you have created. So you want to actually look at those segments. And to do that, you need something referred to as a gel. I'm going to try to stick with the board up, just with the pictures on the diagram here. So this is a gel. A gel is something that you pour. To pour a gel means to take some powder, place it into a liquid, put it in the microwave, and make that powder dissolve. The powder is agarose, which is an extract that comes from seaweed. It's similar to what's found in Jell-O. So I think Jell-O is made the same way. So you mix it up, pour it into a mold, and it solidifies. So what we see is that once the gel is made, on one end of the gel, we have these little holes that are present because of something that's left in there when it solidifies. These are referred to as wells. This is a well, this is a well, this is a well, and this is a well. So into the wells, we place our DNA that has been copied. And remember, we have 1 million copies of this DNA. DNA has a negative charge. And so when we put the DNA into this gel, it's sitting in this chamber. And if you follow the pointer, on this side of the chamber, there's a negative charge. On this side, down at the bottom, there's a positive charge. And so when we turn the electricity on, the DNA moves from this side of the gel all the way down to this side. So it's moving through the gel. We call it migrating through the gel. The speed at which the DNA migrates depends on the size of the DNA. Longer pieces of DNA move shorter, Matthew. They don't move as quickly because they get hung up in the branches of this polysaccharide from seaweed that makes up the gel. Now, let's look at the example that we have from our book here. So we're looking at 2 suspects and blood from a crime scene. What we're doing here is something referred to as short tandem repeat profiling. And short tandem repeats are mutations that occur in the DNA. In fact, they're a point at which the DNA polymerase stuttered. It was producing bases and it said ATG, that's all it was supposed to do, but then it said ATG, ATG, ATG. So each of those is a repeat, a short tandem repeat. They occur one after the other, and they're very short. So if we look at these guys, this one has 12 of these repeats. In other words, ACT, 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 ACT. And over here, on his homologous chromosome, he also has 12. This one has 12 on one of his chromosomes, and he has 16 on his other chromosome. And so what we see is this, that we need to design primers, and we have done so, and they bind to the pink area of the chromosome, copying these repeats. And then we take this DNA and we place it into the gel. So the suspect A goes here, B goes there, crime scene there, crime scene DNA there, and then this is a marker over here I will talk about in just a second. It's something called a ladder. So when we turn the electricity on, look at suspect A. He has 12 repeats there and 12 repeats there. Ben, do these repeats go a long way or not very far relative to the 16?
A long way.
Long way is perfect, Ben. So they're all the way down here. And then look at suspect B. And so suspect B has 2 bands, BANDS. One of them is found right here because it is 12 repeats. It went a long way. And one of them is 16. It's a larger band, so it didn't go quite as far, and it ends up right there. Any questions or comments about why suspect B has 2 bands? Okay, very good. So then, Brooklyn, didn't I ask you a question already? Can't rely on the order here, can I? Spencer, how about you? Are you there? Are you there, Spencer? Sydney, let's go to you. Are you there, Sydney?
Yeah.
Sydney, the question is this. Here's the DNA from the crime scene. And so does it match suspect A or suspect B? See that? Here's the crime scene. Here's suspect A. Here's suspect B.
B.
Yeah, it's definitely B. Very good. It's definitely B. And so that is what you'll be doing in lab this week. And that is really a very simple DNA fingerprint. Now, it's different than a real DNA fingerprint, Kyle, because the FBI uses 16 locations along the chromosome. If one location gives you 2 bands, 16 locations would give you how many bands? 32, right? Would you agree with that? Potentially 32, a maximum of 32, if you're heterozygous for all those. Now, before we leave this, let's talk about the ladder, what's called a ladder, just like a ladder you would climb, L-A-D-D-E-R. It's found over here, and it has a bunch of DNA segments of known length. And so these are probably, let's say, number of repeats. Okay, so this is 2 repeats, 8 repeats, 10 repeats, 24 repeats. And so we can tell the size of these bands using this ladder. So what you see is the 2 repeat band went the farthest because it's the shortest DNA, and then 8, 10, 14, all the way up the board here. This band we know is 12. And so it ran a little farther than the 14. We know this one's 16, and so it ran a little farther than the 18. So that's the value of a ladder. A DNA ladder will always be run with a sample because you want to have some idea of how big those bands are that you observe. Questions or comments there? All right, guys, so here's an example for you of a paternity case. This is how, of course, paternity is determined in humans or in animals. If you have domestic animals and want to do that, it's really not very expensive, at least for animals. I guess there's more. Probably they check it a few times. With humans would be my guess, Sam, but for animals it can be as little as $25 to have this done for you. So what we see is that we have a mother, we have a child, and then we have 2 potential fathers. And so in this case, we can interpret this in the following manner, Ethan. So the child, the bands that they possess must have come from either the mother or the father. So clearly in this case, who's the father? Male 1 or male 2? Male 1 is exactly right. Here's another example of a DNA fingerprint. A very simple one, but we have the ladder found in the middle here. And if we start at the left-hand side, we have the victim's DNA. We have DNA from suspect 1, DNA from suspect 2. And then we have the victim's DNA again, and then scrapings taken from under the victim's fingernails. So who appears to be the guilty party in this case? So Ethan says suspect 1, and I would definitely agree. And so we see that this banding pattern matches suspect 1, doesn't match suspect 2 at all, and it does not match the victim's DNA at all. So that's the value of a DNA fingerprint. Questions or comments there, guys? That's what you'll get to do in your lab this week. It'd be nice if you got to do this in your lab this week, make a transgenic organism. So transgenic organisms are organisms that have DNA from multiple sources combined together. On the screen, on the left-hand side, we have a transgenic axolotl. It's a salamander that has the gene from a glowing jellyfish inserted into it. That gene is referred to as the GFP gene, green fluorescent protein. Because DNA is the same in all living organisms, you can mix and match those without any trouble. If we go over to the right-hand side of the screen, we have a transgenic puppy. And maybe your parents will get you a transgenic puppy for Christmas, Matthew, if you ask nicely. Surely they would get you one. They're only $5,000 or $10,000. And so it contains a gene from, I do think they're sold from the Japanese company. I could be wrong about that. Maybe that's just the pigs. But it's the gene that makes coral glow, a species of coral glow red. And so they called it a ruppy since it's a glowing ruby-colored puppy. So it's important to point this out. It used to cost hundreds of thousands of dollars to make a transgenic organism. You realize that about maybe 4 weeks ago now, the Nobel Prize was given for a technique referred to as CRISPR. I encourage you to read the book Crack and Creation by Jennifer Doudna, one of the 2 recipients, yes, of the Nobel Prize just a few weeks ago for CRISPR. And in it, she says that CRISPR allows us to make genetically modified organisms for tens of dollars. Not hundreds, not thousands, not hundreds of thousands, but like for $1,750, we can make a genetically modified organism. So, I don't know about you all, but that definitely puts it more into the affordable range, Sam, for me, versus, a couple $100,000. So it's amazing the change that's going to occur as a result of that, Matthew, and the ease at which we can make genetically modified organisms today. So here we see transgenic bacteria. Again, we've talked about the process by which we do this, but we point out at this particular, at this point, that products on the market like insulin, I think we said that, the hepatitis B vaccine, tissue plasminogen, and human growth hormone are all produced via this particular process. In other words, you put a gene into bacteria and you let bacteria make the product. And it's the same product that humans make. And so it can easily, be given to humans with no problem whatsoever. You can do other things too. So you can use transgenic bacteria to produce chemical products, which is commonly done, Sam, in industry. You can use transgenic bacteria to aid in plant health. For instance, you could add a bacteria to the roots of plants that produces a toxin, Ethan, that allows those plants to fight off insects on their own. You could also make bacteria, Matthew, that degrade oil. And so like the BP Horizon oil spill back in 2008, maybe 9. So that would have been a good application. I don't think it was ever used there, but these bacteria do exist. We're just always pretty hesitant to deploy something into the environment that we're not absolutely certain is safe and is not going to spread. Now, it is interesting to note how many of the plants that are grown today are transgenic in agriculture, and the answer is almost all of them. And so if we look at the tiny graph that's found in the lower left-hand side of the screen, we go back to 96, which I know predates almost all of you, then there were very few, less than 10% of any crop was transgenic, but this only goes to 2014. And in 2014, 90% of all our major crops are transgenic. So as you look out at a corn field or soybean field, it's almost certainly a transgenic crop. And there are two types of transgenic plants. There are, on one hand, we have plants that are herbicide resistant. And so all the ones we see on this screen, these Roundup Ready plants are resistant to herbicides, specifically glyphosate, which is Roundup. And this is, you'll see this in the spring, even on the hill coming into Wilmore there. So you'll see the whole field get brown, and there'll be little rows of green in the field. And so what they've done is to plant soybeans, spray the whole field with Roundup, and the soybeans grow, everything else dies, and it's a really quick and easy way to accomplish a good crop of soybeans. The other possibility is that there are plants that have the gene for insecticide inserted into them. These are referred to, Kyle, as BT crops, Bacillus thuringiensis, which is a bacterium that produces, Ethan, an insect toxin. Human hormones are also grown in plants. There's a company in Owensboro, Kentucky, a biotech company, that takes the gene for things like, what's the anti-clotting factor? Heparin, human heparin, and puts the gene into tobacco plants, because we know how to grow tobacco plants, and they will grow just fine here in Kentucky. And the harvesting, it's all a lot of work, but it's all worked out. I think they could grow something easier to harvest. And they harvest tobacco plants, and instead of using them for their traditional uses, they extract the hormone from them and sell. It's not actually a hormone; they extract the clotting factor from them and sell that clotting factor. You can also produce antibodies in plants, interestingly, and so the treatment for Ebola, for instance, some of it came from that same company up there in Owensboro. In that outbreak, seems like that was 5 years ago, 7 years ago, something like that. But they were busy working on that. And some of those, although I don't know about the FDA approval and what happened there, but some of it was used experimentally, not experimentally, just in an emergency fashion for one of the Samaritan's Purse doctors, and they believe saved his life. So we go back to transgenic animals for a moment. How do you make a transgenic animal? And so there's different ways of doing it. One possibility is microinjection. In the case of microinjection, you take a very small syringe and you inject your DNA into the early stage embryo, and the DNA incorporates it into that embryo. Another possibility is vortex mixing. In vortex mixing, you take eggs, say frog eggs, and you put them into this little chamber that shakes back and forth very quickly and is lined with needles. The needles just poke little tiny holes. When I say needles, Sam, I mean like specialized, really tiny needles. Pokes very, very small holes in the sides of that egg. It allows the DNA to enter. It's a weird process. It's weird that this happens, but the DNA can be taken up into those eggs or early stage embryos, and a transgenic organism is created. Gene farming is the process used to produce transgenic farm animals that produce pharmaceuticals. Take a look at this. We're taking the gene for human growth hormone, we're micro-injecting it into a goat egg, and we're putting the goat into a surrogate mother. When the goat's born, we got the gene in just the right place. This wouldn't happen automatically, but if we get it in the right place, the goat produces the gene for the human growth hormone protein in its milk. And then, since that's a big accomplishment, we take that goat and we clone it, and so now we have a goat that produces this pharmaceutical product that we need in the milk. It's not widely applied at this point, Ethan, but it is an idea, and there have been prototypes of this made. To the best of my knowledge, it's not used on a broad scale at this point. Veronica, how many animals that you eat are genetically modified?
Almost all of them.
You would think so. That's a good answer based on the plants. But the answer is no, surprisingly. Only salmon in the United States at this point is approved for human consumption. I saw an article in the past couple weeks where a big company that makes those is moving to Mayfield, Kentucky, out in western Kentucky. They're going to do indoor production of these genetically modified salmon. Here are the salmon. This is the genetically modified one, and this is the normal one at the same age. And so interestingly, it's multiple copies of the growth hormone, just salmon growth hormone, but multiple copies have been inserted into this one that causes it to grow much more rapidly. Is this safe to eat? Is this safe to eat? Well, The answer is science would, I wish we had more time to talk about this, but the science would tell us yes. Science would tell us yes, Ethan. And in fact, scientists, this is one of the topics that stresses them out the most, Veronica, because the public, both in the United States and in Europe, I'm a little less certain about some other, some places in the world. It would be interesting to note that. Hopefully they're more progressive, but we're pretty hesitant to accept these things, but science says that they're okay. And so scientists lament that they didn't do a better job explaining this to the public so they would be more accepting of these products. But there's no evidence at this point that there are any issues with these, either for humans or for the environment after thousands of studies. So we're going now to gene therapy as we taper down here to the end. And so gene therapy is the process of modifying the genes in a human being. Is this okay to do? And most people would say yes. I mean, we don't do it just to make someone jump higher or have a different color hair, but you know, if they have a life-threatening disease, then we're willing to give it a shot. So one possibility is doing this ex vivo, so outside the body. In vivo would be inside the body. Children with severe combined immunodeficiency, SCID, lack an enzyme that they need in their white blood cells. And so what can be done is that the gene is placed into a virus, which is shown here. This little girl who has the disease, her blood is removed and the white blood cells are modified outside the body, and then the white blood cells, once the healthy gene has been added via this virus, are placed back inside her. And there's been moderate success with this in treating SCID. Now, you got to remember, CRISPR is going to change all this. So CRISPR is much better than using these viruses to deliver healthy genes. And so there's already at least a trial that's going on, testing, at least one, looking at the possibility of fixing, healing, I suppose, is that the words you would use, someone that has sickle cell anemia by editing their DNA. So thoughts or comments there? With any luck, you know, this says cystic fibrosis. So with any luck, CRISPR will be able to treat and cure cystic fibrosis. We go on now to our last topic in our whole class, which is genomics. Genomics is a study of genomes, and what we see is that the Human Genome Project was completed in 2003, and the goal of the Human Genome Project was to sequence the 3.2 billion bases that make up the human genome. And it cost about $3 billion to do, and I don't think we've reached this quite yet, but for just 100, this one company said that they could sequence a genome for $100 by the end, Matthew, of 2018. I think we're close to that, but maybe not quite there at this particular point. The technology has gotten so much better. So here we have Collins, who we've cited several times, including in our discussion of evolution, Fauci's boss and head of NIH. See him on the news quite a bit these days. Here's Venter. He's certainly still around. He was in charge of the privately funded portion of the Human Genome Project. There's Venter and there's Collins on the front of Time magazine after they finished this massive undertaking. So what they said, though, when they got finished this was that this is just the beginning. So we have all the sequences, but what we need to do now is figure out what these sequences do. One of the ways of doing that is functional and comparative genomics. And so essentially, what's done is this. You look at what genes are expressed. What we have on the screen is something called a microarray. A microarray is a little tiny chip, about 1/2 inch by 1/2 inch, Sam, and on it, there are all these DNA probes. See these? And they're complementary for particular genes. So you extract the RNA from a cell, turn it into complementary DNA, and you spread it out across the slide. If the complement for a gene is present, then it binds based on the rules of complementary base pairing. And if that happens, there's a fluorescent probe hooked up and it lights up. And so we can tell, you have to read it obviously with a computer, Sam, because you can't, with your eye. This is 1/2 inch by 1/2 inch. You're not going to be able to read that. But it tells you which genes are being expressed and which genes are not. And so that's the key. Remember, it's one thing to understand the sequence, to have the sequence of all these genes. And we do have that, Kyle. But it's quite another to understand what these genes actually do. So the final frontier is proteomics. Now, remember what we said on Wednesday, that the overall size of the genome, Matthew, is not a good predictor of the complexity of an organism. We said that there's the example of DSCAM as a result of alternative splicing. One gene and 30,000 bases in fruit fly, 30,000 proteins in fruit flies as a result of alternative splicing. So that's the answer to that question. The proteome is larger than the genome. Why? Well, because of alternative splicing, largely. If you remember that from Wednesday, you can put that RNA together in different ways. And so proteomics we'll use to develop drugs and all sorts of, it will greatly enhance our understanding of human medicine physiology once we obtain those. Guys, we've reached our last slide of the semester, sadly. And so the title of this last slide is bioinformatics. And bioinformatics is the study of and manipulation, of this genetic information. And so this is something that pervades all fields, that is present in all fields today. Central dogma of biology is what? Sam says DNA to RNA to protein, and is certainly exactly right. And so remember, whatever field you're in, whether it's ecology or human physiology or microbiology, knowing the genomes is something, knowing the genes is something that's of great value to you. So whoever you're going to be, a doctor, a physical therapist, a scientist, a teacher, certainly, you're going to need to know some bioinformatics. And so there's an extra credit assignment that's posted for you on bioinformatics. And I want to be clear, the extra credit assignment is a worksheet. And so the goal is to take that worksheet and use these resources and complete that. And then there's a quiz. But I do want to be clear, if you open the quiz and then start on this, you'll never get it done, Kyle. So it'll take you a while to do the worksheet. So you want to have the worksheet all done before you open that quiz. The quiz just if you have the worksheet done, you just go click, 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 and you'll be done. So that's the overall premise. We don't have quite enough time to go over these tools here. So there will be a YouTube video if you're interested in that posted on this last slide and how to use these tools. These are our tools that are provided to us by the NIH through the by the NIH, and so they're available to everyone. They're government-funded resources, and they're the biggest. One of these, GenBank, is the biggest depository of sequences in the world, Matthew. So it's definitely of great value to be able to use these. And so if you're interested in that, then look over the next couple of days. That video will be posted, and you're welcome to ask me questions as you work on the worksheet. So as we close here, I've got a couple short announcements for us all. Let's see. In no particular order, there is another extra credit opportunity posted for you, and it's a chemistry quiz. To be honest, you were supposed to take that chemistry quiz a long time ago, but I didn't ever get it done, Sam. We've never had an online lab before. It usually happens during lab, and so it's optional at this point. It's 10 points extra credit if you'd like to take that, half a point apiece for 20 questions. Honestly, it's probably pretty easy. You can go back and look at chapter 2. If you're taking a chemistry class now, I would imagine that will be quite easy for you. So that's available for you. There's the bioinformatics extra credit. I already mentioned that. And I think the last thing is the final exam. And so the final exam is cumulative, but the new part of the final exam is pretty big this year. So I have a review sheet in front of me right now that's posted on our Discovery site. And what you'll see is this, that the cumulative chapters on the final are 1, 3, 4, 5, 6, 7, 8, and 15, excluding the ecology chapters. I mean, I didn't mention those. Those are not on here. The new material is 9, 10, 11, 12, 13, and 14. That's a big exam. Now, this exam is always 100 questions in length, and so it'll be right about that this time around. This time around, the new material will be a little bit more. The cumulative will be a little bit less. The review sheet has questions, Kyle. And so all of the questions on the cumulative portion of the final should be present here. It's not an absolute promise, Ethan. I mean, I didn't intentionally, change any, but if you can answer these, you should do very well on the cumulative portion of the final. You will notice I don't have 15 on here yet. I will add that and repost, but every other chapter besides 15 is here. The final exam will occur during our finals week after Thanksgiving. It'll be available Monday. I mean, I think you're going to have till Thursday to finish it. If you need it, take till Thursday. There will be a brief period of time on Thursday afternoon, late Thursday afternoon. I won't be available to answer questions, so if you really think you'll have a question, take it before then. I'm sure I'll be around email continuously before that. And then lastly, your lab exam. So there's one lab left. Everything's up for that lab except the lab quiz. There'll be a review sheet posted early next week for the lab exam. And then the lab exam will be multiple choice, and it'll be available the same time as your lecture exam. So any questions or comments, guys? Ethan has a question. Yeah, Ethan asked, where's the extra credit? And so Ethan, it is, so here's B. This is the class that's going on right now. If I move the Zoom people over here, then, well, a couple things. Guys, don't forget, you got to hit the load more button. So there's your, this video, I guess this doesn't help because people that are watching it will see it, but this lecture will be posted as a video at the bottom of those. I think they're all in the assignments category. Is that correct? No, there's your Biology Through the Eyes of Faith 2. Don't forget about that. I didn't even mention that. Here it is, chemistry quiz, which is extra credit. And then here's your bioinformatic extra credit worksheet, and there's a bioinformatic extra credit quiz. So there it is, Ethan. Good question. Other questions, guys? Any questions from Zoom? Matthew has a question, and if you have a question from Zoom, I can't see all of you, so just shout it out. Matthew. You'll have a bunch of time for the final, like, I mean, maybe like, how much time do you want? 3 hours or something like that? Yeah, it'll be a lot. It'll be more than I think you could ever possibly use. I'll look at what your other time limits are, and yeah, it'll be
3 hours would be great.
okay, Ben. Yeah, and I mean, I'm going to say this now, even though I know you all abide by this. It's absolutely closed book. We all realize that, right? So it's of course it's on the honor system, but it's, you know, if it wasn't for COVID, we'd be sitting in this room week after Thanksgiving taking this. So, but yeah, lots of time is no problem whatsoever. Other questions? Be sure to shout them out if you're on Zoom. I can't see all of you. Okay, guys, good job this semester. It's not really appropriate to say goodbye at this point because we've got a lot of stuff left to do. I'll be interacting with you a lot, but do have a good Thanksgiving, and be sure to let me know as you have questions as time goes on here. So nice work.